Right, and we're going to continue on with the digestive system, this time looking at the intestines. So, the small intestine is going to play an important role with digestion and absorption. This is where 90% of the nutrients are absorbed, so that's pretty important. There are three regions: the duodenum, jejunum, and ileum. They have the same four layers that we see continuously going through. The mucosa here is going to have a lot of absorptive cells because of the digestion and absorption function. Goblet cells to produce mucus, some intestinal glands, Paneth cells, they're going to produce lysozyme and enteroendocrine cells, and more of the malt, where you have the mucosa associated lymphatic tissue. So in your submucosa, you've got duodenal glands that are going to secrete your alkaline mucus. The muscularis, the serosa, is going to completely surround this except for a major part of the duodenum. So here again you can see where you've got the small and large intestine. Here is an internal view of the jejunum where you can see all of these circular folds in there, the plicae circularis. So a lot of these folds in here are to help increase the surface area for digestion and absorption. It increases it by about 600-fold in there. You need to have a membrane for things to be absorbed, so the more membrane, the better. So we have the circular folds, permanent ridges of the mucosa and submucosa that do not actually disappear when the intestine fills. Instead, this is going to cause the chyme to spiral as it goes through. The villi are finger-like projections of the mucosa. They're going to have an arterial, venular, and blood capillary, as well as a lacteal. The microvilli are projections of the apical membrane of absorptive cells. That's going to increase the surface area even more. So you've got villi, which are little folds, and you've got folds on the folds. We'll have the brush border with brush border enzymes, and then the villi are covered with simple columnar epithelium that are going to have microvilli. So for the glands, you've got goblet cells that are between the columnar epithelial cells that will secrete mucins. The enteroendocrine cells, they're going to produce some of the intestinal hormones like gastrin, cholecystokinin, and secretin. And Brunner's glands, they're going to produce more mucus when the chyme arrives from the stomach. So looking at those lacteals and the crypts, each of the villus lamina propria is going to have a capillary and nerve supply for absorbing nutrients. So that central lymph capillary called a lacteal is going to be larger than the capillary, so they can absorb larger particles like lipid droplets. You're going to have muscle contractions that will move the villi back and forth to help with absorption and move the lymphatic fluid. The crypts of Lieberkühn are openings of the intestinal glands into the lumen of the base of the villi. This is going to allow brush border enzymes that are integral membrane proteins on the surface of the microvilli to get in that will break down materials in contact with the brush border. So enterokinase is a brush border enzyme that will activate the pancreatic proenzyme trypsinogen. So the small intestine is broken down into segments. The duodenum is 25 centimeters or about 10 inches long. That's going to be the end closest to the stomach. This is your mixing bowl area that's going to receive the chyme and secretions from the pancreas and liver. It has few plicae and small villi here. The duodenal glands are going to produce a lot of mucus and buffers to help neutralize the acidic contents from the stomach so that they don't damage the absorptive surfaces later on. It's activated by the parasympathetic nervous system during the cephalic phase. Then you have the jejunum. It's 2.5 meters or about 8.2 feet long. It's the middle segment. This is where most of the chemical digestion and nutrient absorption comes. And then finally the ileum, that's the final 3.5 meters or about 11.5 feet long. So your duodenal enteroendocrine hormones, your intestinal tract's going to secrete peptide hormones. that will have multiple effects in other areas and in the accessory glandular organs. So secretin, it's going to be released when the chyme arrives, and it will increase secretion of bile and the buffers from the liver and pancreas. Cholecystokinin will be secreted in the duodenum. This is going to be secreted when the chyme contains lipid and partially digested proteins. It's going to help accelerate pancreatic production of the digestive enzymes, trigger bile ejection in pancreatic juices. The gastric inhibitory peptide is secreted when there's fats and carbohydrates. All of these are going to reduce gastric activity, so you want to shut down the gastric activity once things move into the small intestine. The vasoactive intestinal peptide is going to stimulate the secretion of the intestinal glands. It dilates the regional capillaries so that you can remove the absorbed nutrients, and it's going to inhibit acid production in the stomach. So it's going to help move what you do absorb down the line and shut down the stomach. Gastrin from the G cells is going to promote increased stomach motility and stimulates the acid and enzyme production. Enterocrinin is going to be released when the chyme enters the intestine. It's going to stimulate the mucus secretion in the duodenum. So this is showing here one of the intestinal villi, and then this allows you to see where you have the lacteals, the goblet cells, the brush border, and the capillaries. And here you can see the different cells in here, the absorptive cells, the goblet cells, the neuroendocrine cells, and the Paneth cells. So these are some actual microscopic pictures of it where you can see the different cells and layers. So these intestinal juices and brush border enzymes, you're going to have 1 to 2 liters of these produced daily. They'll have water, mucus. They're slightly alkaline, and they're going to provide a liquid medium to help with the absorption. So these brush border enzymes are inserted into the plasma membrane of the absorptive cells, so you can have some enzymatic digestion at the surface rather than just in the lumen. So some examples: alpha dextrinase, maltase, sucrase, lactase, aminopeptidase, dipeptidase, nucleosidase, and phosphatases. So we still do have some mechanical digestion going on here. It's going to be governed by the myenteric plexus. So with the segmentation, it's going to help with localized mixing contractions. It mixes the chyme and brings it into contact with the mucosa. You have migrating motility complexes that are going to be a type of peristalsis. They'll begin in the lower portion of the stomach and push things forward. So chemical digestion that occurs here: carbohydrates. So you've got various enzymes that are going to do this: pancreatic lipase, alpha dextrinase, sucrase, lactase, maltase in the brush border, and it ends up creating monosaccharides, which you're then able to absorb. Proteins will be chemically digested. So trypsin, chymotrypsin, carboxypeptidase, and elastase from the pancreas, aminopeptidase, and dipeptidase. Lipids, pancreatic lipase is the most important one. You'll have emulsification with the bile salts. It's going to increase the surface area. And then nucleic acids with ribonuclease and deoxyribonuclease. So we're going to have carbohydrate digestion, protein digestion, and lipid digestion all occurring in there, as well as the nucleic acids. So when we look at absorption, the monosaccharides are going to be the way that your dietary carbohydrates are absorbed. And this will happen through facilitated diffusion or active transport into the blood. So all that should be left is the indigestible cellulose and fibers left in the feces. So That's an important component of feces, though, as well. A lot of the good bacteria need high fiber amounts in the feces. Your amino acids, dipeptides, and tripeptides, most are absorbed as the amino acids through active transport into the blood. About half come from proteins in the digestive juices and dead mucosal cells. And then lipids are all going to be absorbed by simple diffusion. So the short-chain fatty acids, these are mostly made by bacteria, go into the blood for transport. And those do play a lot of important roles. So those bacteria balance in the gut is very important for that. The long-chain fatty acids and monoglycerides are going to be large and hydrophobic. And then the bile salts will help form micelles to ferry them to absorptive cell surfaces and reform triglycerides in the chylomicrons. So if they're bigger, they get modified a little bit. They're going to leave the cell by exocytosis and enter the lacteals. So they will eventually enter the blood with a protein coat of chylomicron to keep them suspended and separated. So this picture here just illustrates the different things going across and how they move across. And this is showing them going into the blood supply where they're going to join in. So you can see the things that go into the bloodstream, they enter into the blood right away and they go to hepatic portal circulation and then back to the heart. The things that go into the lacteals are going to bypass the hepatic portal circulation and they'll join back in up at the heart. So that's a key difference there. So the electrolytes, they're going to enter epithelial cells by diffusion and secondary active transport. You've got active sodium-potassium pumps here. Chloride, iodide, and nitrate will passively follow. Iron, magnesium, and phosphate will be actively transported. In order to absorb the calcium, it's going to require vitamin D, a parathyroid hormone. The fat-soluble vitamins, they're going to travel in micelles and be absorbed by simple diffusion. The water-soluble vitamins are absorbed by diffusion. And then B12 is going to have to combine with the intrinsic factor before it's transported into the cells. And it will use receptor-mediated endocytosis. So when we look at absorption of water, 9 liters of fluid are dumped every day into the gastrointestinal tract. Your small intestine will reabsorb about 8 liters of that. The large intestine reabsorbs 90% of that last liter. So the vast majority of it comes back. This diagram here just shows you the different areas and the different amounts that are secreted in those different areas. So we have intestinal reflexes that are going to be stimulated by stretching the stomach. The gastroenteric reflex is going to stimulate motility and secretion along the entire small intestine. The gastroileal reflex is going to trigger relaxation of the ileocecal valve, and this will allow the material to move from the small to the large intestine. So the enterogastric reflex is going to be where chyme enters into the small intestine. That slows gastric movement. So the large intestine is a horseshoe shape. It's about 1.5 meters long, 7.5 centimeters wide. It's inferior to the liver and it goes all the way to the end of the ileum to the anus. It's going to frame the small intestine, so it goes around the outside. There's 4 major regions: the cecum, colon, rectum, and anal canal. The ileocecal sphincter is between the small and large intestine. So the colon itself gets divided into 4 areas. The ascending, it's going to have things moving up. Transverse moves them across. Descending moves them down. And then the sigmoid colon is the curvy area at the end. The way I remember which side these things are on is the left side is descending. I think of it as the left side is the leaving side. So your ascending is going to be on the right. The anus is guarded by an internal anal sphincter of smooth muscle that's involuntary, and then you've got the external anal sphincter that's under voluntary control. So what the colon is going to do in the large intestine is help to finish up absorption. You're going to produce certain vitamins and minerals. A lot of that's done by bacteria. And then you form and expel the feces. So you do have to absorb those vitamins and minerals that are produced by the bacteria. And then it's a place to store fecal matter prior to defecation. That's an important thing for us to function with our lives the way we do. If you couldn't hold that, if it just dropped out every 2 or 3 minutes, it would get quite messy. So that ileocecal valve is going to attach the ileum to the medial surface of the cecum. This is going to receive the material from the ileum. It starts the compaction process. It's opened by the gastroileal reflex. The appendix. This is an organ that a lot of times people think has zero purpose. Really, we don't have anything in our body that serves zero purpose. There are things you can live without, but they all do have a purpose whether or not we understand it. So the appendix is a slender, hollow appendage. It's about 9 centimeters long. It's mostly lymphoid nodules, so it's a lymphoid organ. And it's attached to the posteromedial surface of the cecum. So we call the colon the large intestine because of the large diameter, and it does have thinner walls than the small intestine. It's going to form a series of pouches we call the haustra that make it appear segmented. This is going to allow expansion as you go. There's muscles along here. There's 3 longitudinal bands of muscle, the taenia coli, that run along the outer surface. The muscle tone is what's going to create the haustra. So it's almost kind of like an elastic creating some pouching on there. The rectum forms the last 15 centimeters, and it's expandable for temporary fecal storage. When it moves into the rectum, that's going to trigger the urge to defecate. So at the anus, where you have the exit, you have keratinized epidermis like skin. The rest of it is columnar and non-keratinized stratified squamous cells. So we've got the same 4 layers. The mucosa is mostly absorptive and goblet cells. There's no circular folds or villi here, although there are some microvilli. You have the submucosa, the muscularis, so longitudinal muscles that are going to be modified to make those taenia coli and the haustra, and then the serosa. So this picture here is nice to show. You've got the ascending, transverse, and descending colon. You can see the haustra, the little pouches on there. And then this is the taenia coli muscle making those. And this shows an internal picture of it. And then this is just an illustration of the histology. Here is an actual picture. This side would be the lumen, the lamina propria, where you can see those muscles in here, and the 4 layers. So the colon lacks villi, but has plenty of goblet cells. And then you've got some distinctive intestinal glands that are deeper, but the goblet cells are the main thing in here. It does not produce enzymes, but you want to have lubrication to keep the fecal material moving. You do have large lymphoid nodules throughout. So mechanical digestion here, you'll have haustral churning and mass peristalsis. This is going to drive those contents towards the rectum. We're in the final stages of chemical digestion here. A lot of bacterial action. You can ferment some carbohydrates here, produce some of the B vitamins and vitamin K. So no enzymes are secreted in here. With the fermentation, it can produce some gases in here. So we have reabsorption of things like water and vitamins, vitamin K, biotin, and B5. So the bacteria that are in here do play a lot of roles in your health. That could be a whole list of things. It's a much bigger topic than what we can get into in this lecture. But a lot of times it's even the products they secrete that are important for health, not necessarily just the bacteria themselves. So we have some organic waste in here, the urobilinogen and stercobilinogen, bile salts. So they can be transported in the liver and the blood. And then finally you have defecation, which will be triggered by distension of the rectal walls. So the bacteria are going to convert that bilirubin to urobilin and stercobilinogen. So a lot of this is due to exposure to oxygen. The bacteria break down the peptides in feces, so this will create some ammonia. Indole and skatole are nitrogen compounds that help give the odor to feces. Hydrogen sulfide is a gas that's produced that can give a rotten egg type odor. And then the bacteria feed on the indigestible carbohydrates. This is why that fiber is so important. So they can produce flatulence or intestinal gas. So the defecation reflex is going to be how you're going to start the process of eliminating feces. It requires relaxation of the internal and external sphincters in order for that to happen. The reflex will open the internal sphincter and close the external sphincter when the rectum receives the feces. Then the opening of the external sphincter requires conscious effort. So this gives you some conscious control. If you don't listen to what your body is trying to tell you to do, eventually it will build pressure and the external sphincter is going to get forced open. So it's going to leave at some point if you wait too long. So this is just a diagram showing the types of food things for the carbohydrates and then the enzymes and where they are going to act for breaking them down. So you can follow it by nutrient type with this. So that's going to look at the carbohydrates. This is going to look at protein digestion. So again, these are things that we've covered in the notes, but it's just different ways that you can organize it in your mind. And then looking at fat digestion and absorption. This combines it into a single table here. This tells you what ends up in the bloodstream. So you've got the chylomicrons for the fats, the amino acids for the proteins, and monosaccharides from the carbohydrates. And then here we can see where you would have the ion and vitamin absorption, things that will regulate it, so how they're going to move across, whether it be diffusion, co-transport, active transport. So the different vitamins and the ions. So your digestive system is going to break down the physical structure of food. It's going to break it down into the molecules that go into the bloodstream. Those get absorbed by the cells, and they're going to provide the energy to make ATP. The digestive enzymes, they're going to break the molecular bonds in those large organic molecules. It's usually hydrolysis. So the nice thing with the enzymes is a lot of times you can tell what they're going to break down by their names. Carbohydrases for carbohydrates, lipases for the lipids, proteases for the amino acids, nucleases to break down the genetic material, and the nucleotides. So for complex carbohydrate absorption, your salivary and pancreatic enzymes are going to function at a little bit higher pH, 6.7 to 7.5. They can usually withstand a lower pH. So they'll be broken down into disaccharides and trisaccharides. And then the brush border enzymes will fragment those disaccharides and trisaccharides into monosaccharides. So a maltase will split maltose into two glucoses. Sucrase splits sucrose into glucose and fructose, and then lactase will split lactose into glucose and galactose. And then those can be absorbed by facilitated diffusion and co-transport. So if you do not have adequate amounts of lactase, you're not able to break down lactose, and that's what leads to lactose intolerance. So with facilitated diffusion, it's only going to move the molecule or ion individually through the cell membrane. It does not require ATP, and it's going to go with the concentration gradient. Co-transport is going to move more than one molecule or ion. They move in the same direction. They may require ATP, and they can go against a concentration gradient. So this is used to transport glucose and sodium into the intestinal cells. They both have to bind to the carrier protein. For simple sugars and amino acids, they can bring sodium ions with them. So with facilitated diffusion, your simple sugars get transported at the apical surface through transcytosis, so it will diffuse through the cytoplasm, and it reaches the interstitial fluid, and then it diffuses into the capillaries of the villus to go to the liver. For the lipids, you're going to have that lingual lipase that's going to come from the tongue to make soluble enzymes, pancreatic lipase from the pancreas. It's going to only work on the exposed surface of the lipid drops. It breaks off two fatty acids and leaves a monoglyceride plus a fatty acid. The bile salts help improve your chemical digestion here. It will emulsify them. Lipid absorption. Your intestinal cells will absorb the glycerol and fatty acids. They can make new triglycerides and monoglycerides and fatty acids. The triglycerides and other absorbed molecules get coated with proteins to make your chylomicrons. The intestinal cells secrete the chylomicrons into the interstitial fluid through exocytosis, and then they enter the lacteals and go into the circulation, finally to the liver, and through the system once. So they do have the chance to be stored other places, though, before they get to the liver. The protein digestion, this is more complex and time-consuming. It mechanically starts in the oral cavity, then in the stomach, with the hydrochloric acid beginning it, starting some proteolytic enzymes. Pepsin is going to work on it. When chyme enters into the duodenum, your enterokinase from the small intestine will trigger the conversion of trypsin to trypsin. And then your pH gets adjusted to 7 to 8, and your proteases begin to work. Amino acid absorption, your dipeptidases, they're going to be brush border enzymes on the surface of the small intestine. After they diffuse into the basal surface, the amino acids are released into the interstitial fluid by facilitated diffusion and co-transport. We have a lot of water absorption that occurs here. Cells cannot actively or secrete water absorption. So it's going to move passively down the osmotic gradient. Ion absorption. Osmosis does not distinguish among the solutes, so it will be determined by the total concentration of the solutes. So in order to maintain proper homeostasis, though, the concentrations of some of the specific ions need to be regulated. So your sodium absorption is going to be increased by aldosterone. Calcium absorption is going to be active transport. This is going to be increased by parathyroid hormone and calcitriol. Potassium ion concentration increases as the other solutes move out. And then other ions diffuse into the epithelial cells along the concentration gradient. So vitamins, they are organic compounds. They're required in small quantities. We have two categories: the lipid or fat soluble are vitamins A, D, E, and K. And then your water soluble vitamins are all of the B vitamins, C vitamin, and then you have all but one of these are easily able to diffuse across the digestive epithelium. B12 is that difficult one for digestion. So with cholesterol, when you have your lipoproteins, we call them that because they have both lipid and protein. The chylomicron is an ultra low density lipoprotein that's going to have triglyceride, phospholipids, cholesterol, and protein. These transport dietary lipids from the intestines to other locations in the body. The VLDLs are called that for very low density lipoprotein. They're going to deliver and deposit lipids around the body. Once they drop off some of that lipid, they become a low density lipoprotein, or LDL. These are considered a negative indicator for health because they can build up in the arteries, and they're an indication that you're making a lot of deposits. HDL, good cholesterol, it's going to absorb cholesterol and carry it back to the liver. It's going to lower the risk for cardiovascular disease and stroke. So the mycelials are spherical structures with the nonpolar tails. They're amphipathic molecules on the inside, so they're shielded from water by the polar heads on the outside. These are important in fat and vitamin absorption in the intestines. So that is the digestive system.